I would like to introduce Matthew Kunicki, CEO and founder of CytIQ. He's an expert in flow cytometry and has worked with companies on the academic side and larger pharma. Welcome to the call, Matthew.
On mute.
On mute.
On mute.
Classic mistake. Wonderful. Nice to meet you, everyone, and happy to see so many familiar names in the audience.
Yeah, it's fantastic. We got a great audience today. So Matthew, wanted to maybe start off with a little bit of, can you share a little bit of your background and your experience with working with solutions like OMIC?
Yeah, I think it's really important to take into consideration that I started with mass cytometry. For some of those who are on the call, they recognize that term and they shudder. But I think that it's more that you have to deeply understand the data quality coming off that machine in a high parameter space. And that's my background, as I've been working in flow cytometry and SRL labs, both in academia and in industry, and we utilize a lot of different flow cytometers, spectral, mass, or conventional, in order to support, several different scientific programs agnostically, kind of like how Dotmatics, agnostically helps out so many of us in our data analysis and management.
So Matthew, when you think about, kind of flow cytometry and some of the larger pharma that you were previously working at, what were some of the specific workflows that maybe you were hoping to leverage and address with a solution like Luma Lab Connect?
Oh man, speaking generally, of course, I think that what was a really stressful part of the job in the SRL is trying to help so many different kinds of scientists. And I don't mean the people, but I mean the projects and the particular questions they had. And so being able to look at all of those different projects and manage them equally and supporting every staff or every team member in the way that they need starts with understanding the data. I mean, I've been looking at some of the questions from our audience, and there's definitely some who understand that there's a lot of caveats with the scientific data generation process, not just how we manage it and analyze it. And I think that looking for tools that are already geared towards adopting all these different types of structures and then making it standardized was one of the biggest assets that attracted me to your portfolio.
Whether you mentioned the importance of data, and like you said, a lot of the questions are coming through around questions for that. Were there some example data challenges that maybe you could highlight for the audience today without giving away too much?
Well, one of them we all face is that, and this is specific to flow cytometry, I think more so than others, is all the different types of ways you could write a standardized file. And what I'm talking about is more or less, the number of manufacturers in our space that generate FCS files specifically for flow cytometry. And when you need to parse those, it really, you need a software that's agnostic to all those small deviations between each one of the manufacturers. And I think, in my experience, I've seen a lot of speed in processing all those different data nuances, utilizing a tool that already is aware or can read all of those different data types in a standardized way. So specifically, the multiple manufacturers, and then of course, if you're working in on your own software tool, a custom solution on site, having the staff to manage that, update it, and deal with new features as your scientists come up with new ideas year after year. It just becomes a large headache to manage all of that data, even within our domain specific area of flow.
Yeah, you bring up an important point. When you think about after you selected like the Luma Lab Connect solution, data quality improvements in some of those other areas. I mean, were there some general success criteria that you wanted to establish as a foundation? Because you mentioned, data quality in various instruments, is kind of part of your part of the importance of, kind of looking and selecting a solution like Luma.
Yeah, what is the value add? I mean, clearly what we were doing before, and I say like 10 years, let's just imagine we're all in our SRL shared resource lab or flow core. And you are wondering, how can I do this job better? And I think one of the first things you think of is you can't really hire more staff. So you have to work with reducing the time of analyzing and managing that data, reducing the time of communicating to these different scientists. I think that one of the biggest value adds that we have is being able to just manage the business. So if you are tracking in data from across a bunch of different sites or a bunch of different studies or a bunch of different instruments, as I mentioned, being able to see which project teams are using those tools or understanding, how is it that we could best utilize this specific flow cytometer for sections of our entire business? And again, speaking generally here, I think that you really need to appreciate that this is a tool that's hitting like the core infrastructures of our science. It's not just, a flashy way of doing biomarker discovery, but it's about how do we work again with all these different teams in a way that can actually get us to the same results faster. And when you do that, the value add becomes very clear. And I think we're excited to see is that these are sometimes the first stepping stones, as you imagine, to aspirational goals. Where now that we're more fluid with the science that we're doing today, what can we really explore with that high parameter space that we've been learning from for the past 10 years?
So I know it's really early days from the Luma Lab Connect deployment, and as you mentioned, time savings, quality, other areas. But are there some general positive outcomes that you believe the team or trends, I should say, that the team was starting to see with the deployment of Luma Lab Connect?
Oh, I was kind of hinting at this before, but it really reminds me of a good mentor of mine that I've been working with for a few years now. He shared how he was one of the first ones to bring in Microsoft Excel to drug development. And I was looking at him peculiarly about, thinking that this is such a large tool, Microsoft Excel, everyone knows it. And how is it that you specifically brought this in? And he shared a story about how there was a time when investigational new drugs were written by hand, or at least some of the data was written by hand and recorded by hand. And so for the very first time, you have all these scientists that are asking, resources to manage all that handwritten data, unstructured data, as we call it. And here's a new scientist, early 90s, maybe a couple years after college, saying, have you ever thought about using Microsoft Excel to collect Western data?